So our second speaker in this session would be Dr. Taichi Suzuki. Dr. Suzuki is a postdoctoral researcher in the Department of Microbiome Science at the Max Planck Institute of Developmental Biology, working in Ruth Lei's lab. His research focuses on studying host-associated microbial community ecology and evolution, mainly in the guts of wild mice and humans. He will talk to us today about the codiversification of gut microbiota with humans. There you go, Taichi.
Yeah, thanks for the introduction, Noah. Can you hear me fine?
Yes. Yeah.
Good. And thanks again for the organizers for this great symposium. I really enjoyed all the talks, especially all these strain level analysis, which will relate to this talk. And I also want to thank the sponsors that are making this event free. I think it's a great model that really supports this field. So thank you as a researcher in this field. And today I'm very excited to present our work and to represent this truly collaborative team that includes researchers inside and outside the Lei Lab, which I'll be introducing throughout the talk. And the work that I will be presenting today got just published in bioRxiv as of yesterday. So it's a brand new work, and I hope you'll all enjoy the story of how we try to identify human gut microbes that may have passed on from our ancestors. So as you know, many microbes live inside and outside our body, and some of those microbes do affect our normal development and health. And we also learned that these important microbes or the communities can change based on a variety of factors like diet, age, or host genetics. But for this talk, I'd like to ask a pretty fundamental question. Where did our gut microbiotas come from in the first place? So this is asking about the evolutionary origins of the human microbiome. So this is a map showing the participants of the symposium. This is already a week old, but it's quite impressive that we're truly an international group. And based on what we know, we likely carry different strains based on where you live. So did you acquire your strain from your local food or local environment? Or did you acquire those strains from your parents or grandparents or the ancestors that first settled in your country? So these type of the questions that I'll try and answer today. So just to back up and make sure that everyone's on the same page, there's two types of transmission, vertical transmission and horizontal transmission. So vertical transmission is where the transmission happens from parents to the offspring. And horizontal transmission is a transmission between unrelated individuals, or sometimes extended to include these acquisition of microbes from the environment. And we've heard this topic, but I'll just cover again that at least in mammals, we're born mostly sterile, and mostly meaning people have identified microbes from the placenta or blood, but majority of the microbes do come from the initial colonizers come from the mom, so through the vaginal canal and skin and breast milk and so on. And later in life, you get microbes from the diet and other individuals. And we also know these initial colonizers are important for health because they can dictate the community assembly processes later on, known as priority effects that was mentioned in many talks today. And just to add another layer, these transmission modes differ by microbial taxa as well. So this is the work I did with Andrew Moller. He's currently assistant professor at Cornell University. And this is the work done in the Nachman lab at UC Berkeley that we collected wild mice from two populations and created inbred lines in captivity, keeping them over 10 generations and over 3 years. So using this data set, we tried to study the transmission modes of the bacteria in mice. So you can see, let's see. If you take a transmission index or score as a x-axis, you can see that distribution is not normal. You can see this bimodal distribution. And what this means is that some microbes are more specialized in vertical transmission, which tend to be anaerobes that cannot live under oxygen. And microbes that seems to specialize in horizontal transmission tend to be aerobes that can live under oxygen and fly around the cages. And these included human pathogens as well. So if there are these microbes that specialize in vertical transmission for many generations in mice, can we find this in humans? And this is a map that's showing ancient dispersal patterns of modern humans in the past 100,000 years. So as we know, we originated in Africa and spread across the world, and we established genetically distinct populations. So if there really are these microbes that are transmitting over thousands of generations, can we identify microbes that show a similar migration pattern as of humans? And Helicobacter pylori may be the most famous and maybe the only examples that show this matching patterns of human migration. So they live in the stomach or the upper gastrointestinal tract, and people have isolated these strains and genotyped them. So the upper panel is showing you the nucleotide diversity of H. pylori, and the bottom panel is showing the inferred migration history of the bacteria, which matches the human migration pattern. So this is a quite impressive work, and we were wondering, besides H. pylori, can we identify other gut microbes that may have co-diversified with humans among the thousands of species that we know? So one approach to get to this question is to test for co-phylogeny. So this is to compare the host phylogeny and the microbial phylogeny and look for congruence. So for example, if you look at the very left panel, if you see this matching pattern, it's called codiversification or co-phylogeny. And please don't get confused. So this is a pattern, and there are a lot of mechanisms that can drive this pattern, and one is vertical transmission. And I don't want you to be confused by another concept called phylosymbiosis, which people make community level bacterial phylogenies and compare it to host tree. But here we're looking at individual lineages of strains. So you can think of it like a species tree of a bacteria. And in contrast, in the other extreme, if you see a pattern like this, like when you see a lot of mismatches, this suggests there's a frequent exchange of strains between the populations. So this is consistent with horizontal transmission. So the whole idea here is to try to rank the human gut microbiome, microbial species, by their degree of co-phylogeny. And people have done this. So Nick Youngblood in our lab showed how mammals show the strongest signals of codiversification with gut microbial species among vertebrates. So he compared fish, amphibians, reptile, birds, and mammalian hosts, and you can see the mammals show the strongest signal of co-phylogeny. And perhaps this is due to some mammal-specific traits like milk production or like extensive parental care. And this work was done using a single gene, 16S ribosomal gene. So that gene is short and you cannot go down to a species level, so the taxonomic resolution is limited. And within mammals, Andrew Moeller showed how hominids show signs of co-diversification with bacteria. So on the left, we have this host tree. On the right, we have the bacterial tree showing the Bifidobacteriaceae family that includes Bifidobacterium genus, which is famous for mom-child strain sharing. And this work was also done using a single gene, gyrase B. And this gene is longer, so you can have a better resolution, but this can only assess certain families of bacteria. So it's limited to what taxa you can survey. And the obvious question is this co-phylogeny signal extending within human populations? And this has not been tested. But at least we know that human populations have different strains of specific gut microbial species, like more of these recent methods like StrainFlan, developed by the Segata lab. So these methods are based on multiple marker genes from extracting from the metagenome. So the basic idea is to try to extract species-specific markers from the metagenome and try to study the strain diversity. So this is focusing on the most dominant strain per species, so per individual, right? So for example, for Bifidobacter longium, this is a global strain diversity, and you can see certain strains are dominating in certain populations. But to really test for co-phylogeny, you need matching sets of human genomes and gut metagenomes. And this has been lacking in the field. So we went out and collected these paired data sets from Germany, Gabon, and Vietnam, mostly recruiting mother and child pairs. And this field work was led by Victor, Liam, and Mirbu from the lab. And through collaboration, we added samples from United Kingdom and Cameroon. So Tim Spector provided the UK samples, and Laura and Ran provided the Cameroon samples. So at the end, we have 747 adults with human genome and fecal metagenome, and their 386 children for some of the mothers. So this is the data set that we'll be using. And here's some results. So this is the human phylogeny from our data set. And you see expected pattern. So you can see the Asians and Europeans are being a sister clade that's nested within Africans, as you expect. And one thing to point out is these hunter-gatherer population that we collected from Cameroon and Gabon, sometimes called pygmies, that they're living in a geographically distant location, but they're each other's closest relatives. So I'll come back to this in a second. So this is the human tree, and now let's look at the bacterial tree. So some bacteria looks like these that look really similar to the human tree. So for example, Roseberia hominis and Collinsella aerofaciens, you can see the most basal strains tend to be African strains, followed by European strains, and then Asian strains. And you can see a similar pattern in Prevotera cochlea and Eubacteria rectale. The pattern looks more complex, but if you look at each subclade, you can see the African strains tend to be more ancestral, and Asian strains and European strains tend to be more derived, consistent with the human phylogeny. And just to point out those hunter-gatherer samples, so their strains are each other's closest relatives, even they're living in a different place. So this supports vertical transmission and argues against environmental acquisition from their local soil, for example. So these are the top taxa I'm showing you now, but many of the taxa actually looks like these kind of parabacteroids and bacteroides have this multicolor tree, all mixed match. So there's not much pattern here. So this is again consistent with more frequent exchange of these strains between these populations. So we can keep staring at these trees, but we can run statistics too. So we try to rank the taxa by using statistics. So I'll just briefly explain these tests. So we applied 3 different types of tests on 2 types of trees. So these co-phylogeny tests are permutation-based tests that you're comparing different phylogenetic distances between 2 trees, the observed tree and the permutated tree. So that's how we're getting the p-values. And the best tree is we're using the best maximum likelihood tree. And the consensus tree is where we're collapsing the branches with low bootstrap support. So at the end, we ran 6 different tests. And if all the 6 were significant, we gave a co-phylogeny index of 6. And when you ran 6 tests and nothing was significant, then we gave an index of 0. So this is how we ranked the taxa. And when you look at the taxonomy, it's pretty interesting that the firmicutes, species in firmicutes show stronger evidence of co-phylogeny compared with Bacteroidetes. And this is consistent with other studies that found species or strains in firmicutes tend to be more population specific. And also this makes ecological sense too, where when you look at the bottom taxa, these species are used for markers for fecal pollution in rivers. So there's some experimental evidence that these taxa can survive and be dormant in water. So perhaps those are the ways that can transfer or transmit between populations. And what was very surprising is this discovery by Kelsey Huss, a postdoc in the lab, that showed that the greater evidence of co-phylogeny, the smaller the genome size. And this is very exciting because in like insect literature, this reduction of genome size is associated with intimate symbiosis between the host and microbes. So here we were a bit worried that this correlation was driven by the genome size difference between the phyla. So we tested within each genera, and you can see that the 7 out of 8 genera showing the expected pattern. So that's significant based on binomial test. So I think, or we think this is the first evidence in mammals that shows this reduction in genome size with greater association with the host. So next, we tested co-phylogeny within population separately. So for example, if you look at these top taxa, they were significant using all populations, but when you make the trees based on only African samples or Asian samples, you see the significant signal extend. So I think this is the strongest evidence of this study that I'm supporting the vertical transmission, because say if there was a dietary factor that's driving this co-phylogeny pattern between populations, we don't necessarily expect the same dietary differences driving the co-phylogeny pattern within populations, right? So that's arguing against these environmental acquisitions from the environment, at least in these top taxa. So next, we tried to quantify the strain transfer events using stochastic character mapping, and the results are consistent with the out of Africa migration pattern. So here, the each dots are showing the microbial taxa, and the greater the evidence of co-phylogeny, there's a greater proportion of Africa to the rest of the population transfer events. So I'll just explain this a bit. So here, and you can see that ancestral state seems like an African strain, and then it got transferred to Asian population and European population. So if you calculate all the transfer events, there's a greater proportion of Africa to the other population transfer events. And the opposite is true for the bacteria that show least evidence of co-phylogeny. Right? That's why they have the low value. So you can do this for all the major human groups. And you can see that Asian to the rest transfer events didn't predict the co-phylogeny index, but Europe to other transfer events, you can see this pattern that the taxa or strains that's distributed globally may have been transmitted from Europeans to the rest of the population. So overall, this is consistent with our evolutionary history. And so far, I've shown you results from 5 populations. So how general are our findings? So to get to this, we added additional public metagenomes, including more than 1,000 human metagenomes from 19 countries and more than 100 metagenomes from 27 wild primate species. And we compared these strain transferring events between the top 10 and the bottom 10 taxa. So here I'm showing you some examples from the top 10 taxa that included the primate strains. So just to orient you first, these branch colors are corresponding to more coarse genetic structure, shown here. And the outer ring is corresponding to more fine genetic structure. So first, you can see that overall, the colors are clustering to each other, showing that there's some population-specific strains, and our initial results with the 5 population was pretty robust. And then you can see that the when you root the tree by midpoint, you can see the primate strains tend to be the most basal strain, followed by the African strain, so on. So again, this is consistent with our evolutionary history. So this is showing the top 10 taxa, but when you look at the bottom 10 taxa, you see this random pattern again. So the colors are mixed and there's not much pattern. And these primate and African strains are not the most basal group, and they're scattered across the tree. So again, this suggests these transfer events between these populations or even between species. And we also quantify the strain transfer events. And this is the result that I already showed with the 5 population. And it's just showing you in a different way. So when you compare the top 10 taxa to the bottom 10 taxa, you can see there's Africa to rest transfer events are more common in the top 10 taxa. And after you add the public metagenomes, you see the same pattern. So again, this supports the initial finding. And I haven't shown you the results yet, but when you look at the children samples, you can see the same pattern. It's marginally not significant if you don't exclude the outlier, but overall, you can see this pattern that's consistent with an African origin for the top taxa. And when you look at the list or the ranking of the child taxa list, it's dominated by Bifidobacterium species. So these are the 4 species that we tested. There's no clear basal group, but they tend to cluster by population. And when you look at the ranking, you can see all the 4 Bifidobacterium species were significant. And this signal extended within population as well, again, supporting the role of vertical transmission. And you might be wondering that the taxa that I showed for adults will likely show up in the child, right, if it was vertically transmitted. But because the compositional difference between adult and child is so large that we couldn't test many of the same taxa in adult and children. But regardless of this power issue, we found that the Prevotella copri showed evidence of co-phylogeny in both adults and children. So again, this supports this role of vertical transmission. But still, this is a pretty indirect way to infer this. So we tested this directly by looking at strain transferring events between mother and child. So this part of the work was led by Hagai and Liam. Hagai is a postdoc in the lab that he developed this tool called SynTracker. So this is using the long stretches of DNA and look for synteny. And that's how you're comparing the 2 genomes. And I'm looking at the relatedness. And Liam used InStrain, which is a state-of-the-art tool to look at the strain diversity developed by Jill Banfield's lab. So this is using a population-level nucleotide identity. So by using 2 very different methods, you find similar results. And you can see a lot of Prevotella species showing up as significant. So what it's showing here is that, say, Prevotella strains between related mother-child pair is more similar compared to unrelated mother-child pairs, as you expect. So this was reassuring. And when you think about the drivers of these codiversification pattern, it's likely that it's not only the strict vertical transmission from mother to child, but it also possible with this within village transmissions. So this is the results from InStrain. And these blue bars are showing the strain transferring events within the mom and their own child. And the gray bars and the gray numbers are showing the strain transferring events between mother and unrelated child. So if you look at Gabon, you can see the majority of the transferring events are happening between mother and the own kid. But in the same town, you can see strain transferring events from mother and unrelated children. And in case of Gabon, this happens even between villages or between towns. In the opposite end, in Germany, we only get to sample 1 location, but we didn't see any strain transferring events within the same town, unlike Gabon and Vietnam. So this result may be driven by the hospital system, sanitation differences. Can't explain these patterns. But I think this bottom message of this slide is to show how not only strict vertical transmission generating this pattern, it likely includes these transmission between individuals that live in close proximity can be driving these patterns as well. So in summary, we provided a list of gut microbial taxa that show evidence of codiversification with humans. And we supported this result by adding additional public metagenomes from other human populations and primate species, and also using multiple methods to try to get to this mother-child strain sharing. So I think overall, this results support long-term vertical transmission for thousands of generations, generating at least some of these patterns. So I think the next obvious question is to look for functional differences between these co-diversified strains and think about the roles they play and how they impact our human health and evolution. So I would like to close this talk by, this is the last slide, just to think about the roles of co-diversified taxa in human health and evolution. So when you think about a local adaptation or adaptive evolution in general, I think humans are one of the most studied systems that's out there, right? Human geneticists and evolutionary biologists have identified all these genes and traits that are under positive selection. For example, adaptation to new diet, new climate, or new pathogens. And all of these classic examples are associated with differences in microbial composition. And the most interesting ones are even adaptive alleles can predict the relative abundance of the bacteria that performs a similar function, like producing similar enzymes. So what this means is that if there's a bacteria that performs a similar function as your adaptive allele, those individuals without the beneficial allele can survive, right? If you have the beneficial bacteria. So this can slow down the process of genetic adaptation because it'll slow down the spread of these adaptive allele. And in contrast, if you have a bacteria that facilitates your adaptive allele, then you can further speed up adaptation. So microbiome may slow down or speed up host adaptation, and these population-specific strain or co-diversified strains can be involved in these processes. So I'm happy to discuss these type of ideas after the talk as well. So thanks again for listening, and this work was funded by the Max Planck Society, and I'd like to thank all the people who are involved in the study. So thanks again, and I'm happy to answer questions.